Welcome to my pitch for the Biomedical Delta Thesis Awards 2020. I'm Inés Beekers, and I would like to present to you the work I did during my PhD at the Department of Biomedical Engineering from the Erasmus Medical Center. My thesis is entitled Local Drug Delivery by Microbubbles, and I'll defend my dissertation on July 8, 2020. Before I go into the details of this project, I would like to give some background knowledge to present to you the aim of my PhD and why it was so interesting to investigate. So let me start in the beginning. In our bodies, we have our vessels, and these vessels can supply oxygen and nutrients throughout the entire tissue. And these vessels are all lined with endothelial cells, which form a great barrier, which is of course very important because we don't want blood leaking out everywhere, right? However, the barrier that they form also proves to be a major challenge for drug delivery. Because we administer drugs intravascularly, and then they flow throughout the entire body and have a very hard time actually getting out of the vessel to the tissue behind it. We have a solution for this: microbubbles. Microbubbles are gas bubbles of about 5 micrometers, the size of a red blood cell, and they are encapsulated in a lipid shell that allows them to be stable in the blood flow. We can actually target these microbubbles to receptors expressed specifically by diseased cells, so that microbubbles are only located in the tissue we actually need them to be. What is very interesting is that we can apply ultrasound, and ultrasound makes microbubbles oscillate, so they grow big and small due to the ultrasound wave. If you co-administer drugs with these microbubbles and you apply your ultrasound, the microbubble oscillation will cause uptake of drugs. The drug uptake is known to occur through 3 pathways. We know that we can create pores in the cell membrane such that actually drugs are able to get into the cells. We also know that we can cause opening of cell-cell junctions such that the cells open up and let drugs through to the tissue behind it, like you can see over here. And we even know that we can stimulate active uptake through endocytosis. We know these 3 pathways exist, but it remains a challenge to understand which microbubble behavior we need exactly to predict which cellular response we'll get. And we actually need a better understanding of these underlying mechanisms of microbubble-mediated drug delivery to be able to aid clinical translation of this technique and the therapeutic potential be fully used. So actually what I'm saying is that we want to see how a microbubble is oscillating, and to do that we need a special camera. Because these microbubbles in an ultrasound field, they oscillate about millions of times per second. So you need a nanosecond temporal resolution to be able to see a bubble moving. To do that, we luckily at the Erasmus MC have a very special and unique camera called the Branderis 128, which is able to record 25 million frames per second. So we can see how a microbubble oscillates when you put ultrasound on. On top of that, we want to see the cellular response. We want to see how a cell responds to this micro oscillation, and ideally to do that you need a confocal microscope because it has nanometer spatial resolution and it allows you to use fluorescent dyes to stain whatever you want. And up to now, or up to my PhD, we could either study the micro oscillation or the cellular response, but we were never really able to do both at the same time. So in order to do that, we developed a novel optical imaging system. Together with Nikon, we have a custom confocal microscope that was coupled to the Branderis 128. And with this unique system, we can now, for the first time, study both the microbubble and the cellular response in the same field of view simultaneously. Apart from this methodological innovation, I also worked on ultrasound-compatible cell culture devices. So we can culture our cells in cell culture devices, but we need to make sure that we can get our ultrasound field nicely into those devices such that we can insonify the microbubbles in a controlled manner. Therefore, I investigated how microbubbles oscillate in a 2D monolayer device called the CliniCell, and I also investigated the pressure field distribution in a 3D vessel on a chip device, the organoblade. I investigated how the pressure field looks like in these microfluidic channels using simulations, and also by using the bubbles as little pressure sensors. So by using the novel optical imaging system and an ultrasound-compatible cell culture device, we can study the microbubble cell response. So here I will show you 2 examples of these studies. So this is a monolayer of endothelial cells before ultrasound, and we stain their nuclei in blue and the cell membranes are stained in green. And here there's one single microbubble located in this field of view. In the medium of these cells, we have added propydium iodide, PI. And PI is a drug that's very ideal to track membrane perforation, to see if we create a pore in the cell membrane. Because what happens is as soon as you create a pore in the membrane, PI is able to go into the cell, bind to DNA and RNA, and the cell will turn red. So you know very locally and exactly where you create a pore in the cell membrane. So this is the field of view before ultrasound, and during ultrasound we can record this microbubble oscillation using the Branderis camera, and we can also quantify this oscillation so we know how much the radius is changing as a function of time. But we are of course very interested in what does the cell do when we have this microbubble behavior. So we can see only like 3 seconds already after this oscillation, we see very local uptake of PI, the red dye, flowing into the cell, which means that we created a pore in the cell membrane. And over time, you can actually see this cell filling up with the red dye. I'll play it as a movie. So I hope you can appreciate how local the drug delivery is that we create. There's really one pore formed at the microlocation and the red dye flows in, fills up the cell. We can locally deliver drugs to the cells. We can not only study sonoporation, but we can also study other underlying mechanisms that are going on. And I'll show one example, which is the intracellular calcium fluctuations that are caused by oscillating microbubbles. So again, we have before ultrasound, a field of view that shows a monolayer of endothelial cells, and the arrow points to the location of one microbubble. So we know how the cells look beforehand, and we can, during ultrasound again, record the microbubble oscillation and quantify it as a function of time. So we know exactly how the bubble is oscillating to create that cellular response. If we then look at how the cell is responding, we see 3 things in this movie that are very interesting. First of all, we can see the local drug delivery of propydium iodide occurring in this cell. So we create a membrane a pore in the membrane of that cell, and red dye is able to flow in. So we sonoperated the cell. Next, we see this huge increase in white dye, which means that the intracellular concentration of calcium increases enormously when we sonoperate. And this not only occurs in the cell that you sonoperate, but it's actually also transmitted to all the surrounding neighboring cells, which is called a calcium wave. So actually we are able to affect all the neighboring cells as well by this single microbubble oscillating on one cell. And thirdly, what we also see is the opening of cell-cell contacts. So the cell is very nicely attached to its neighbors in the beginning, before ultrasound, and once we apply ultrasound, you see the attachment of these cells from its neighbors. It's like retracting and opening up. This means that drugs are able to go out and treat the tissue behind it. So to sum up, we have studied microbubble-cell interaction, and we have observed that higher microbubble excursions cause sonoporation, especially when the excursion is higher than 0.75. When we have sonoporation, we also get an intracellular calcium fluctuation. So we cause the calcium in the cell and in the neighboring cells through a calcium wave to increase. And we quantified these calcium waves to propagate at 15 micrometers per second. On top of that, the intracellular calcium fluctuation can be reversible or irreversible. And when it's irreversible, the excursion amplitudes of the microbubble were higher than 1 micrometer. And these irreversible calcium fluctuations actually very nicely correlated with retraction, with these opening of cell-cell contacts that allows for extravascular drug delivery. We not only investigated this in 2D monolayers, but we also investigated the 3D vessel on a chip by using the organoplate by Biomedical. And we actually were very nicely able to develop a protocol to culture microvessels of endothelial cells in the microfluidic channels to study microbubble-mediated drug delivery. I hope you enjoyed my pitch. Thank you for your attention.